I'm going to talk today about the whole spectrum of RV521, so it's quite a long story. I'll try and make it as quick as possible. I want to talk about an introduction to ReViral. It's a slightly different talk to what you've heard, I suppose. I'm going to talk a little bit about the company. It's a company most people, if not everybody, hasn't heard of. A little bit about the disease. I don't need to talk about that. There's too many RSV people here. And then a bit discussion really about how we progressed our initial idea into a compound with some clinical data, which I'll finish up with. Okay, so my declaration is I'm a founder of ReViral. And myself and a group of friends back in 2011 got together, put our redundancy payments together, literally, and started ReViral. It's a company founded out of what was Arrow Therapeutics. So people might have heard of Arrow from the dim and distant past. We founded it and we initiated our fusion project really as our founding project for the company. So really up to this point in time, RV521 is ReViral, I suppose. That was way back in 2011, and we moved through various rounds of funding, originally initiated by a Wellcome Trust Seeding Drug Discovery Initiative funding, which actually saved our bacon, quite frankly. Funding in the UK is difficult. It's a recession-hit funding background and is probably going to become a Brexit-hit funding background, I'm afraid. And it'll probably only just get worse, as is everything, I suppose. The compound and the company then moved through various fundings, angel funding followed by a couple of rounds of venture capital funding, which have allowed us to move into clinical studies as the spending starts to get far more serious, obviously. Now, RSV, as we know, it's really a cousin in many ways, an unknown cousin of influenza. Most people haven't heard of it. It's a leading cause of lower respiratory tract infection. It results in many hospitalizations, and its key populations are children up to the age of 2, and there's no effective antiviral treatment currently in existence, with Synagis certainly in Europe used in far lower terms than perhaps it is in the UK, in the US. We looked at targeting fusion, and we looked at compounds that have a mechanism whereby we jam the pre-hairpin intermediate. So the blue arrow shows the sort of complex that's formed as RSV approaches a target cell. The fusion protein is activated to a pre-hairpin, and this is jammed. And the structural information which has appeared on this target is shown at the far right, and this basically describes the sort of binding site location for fusion inhibitors of the RSV fusion protein. I'm a medicinal chemist, so one of the most important things to me after the disease and the target, obviously, is how we're going to test the compound. We tended to use a make, design, test cycle, so we worked through this cycle as many times as we could, synthesizing the compound. And this cycle was really based on potency and pharmacokinetic evaluation. So we worked through potency, fusion cell assay through plaque assays, cytotox, into in vitro ADME and in vivo ADME. And compounds that really met some sort of requirement at that stage span out into a whole range of virological assays, really, working through from time of addition studies, mutant generation studies, through to models specifically of infection, either in the human air epithelial or a mouse model in this case. And compounds that actually came out of that with a reasonable profile would move into preclinical and clinical evaluation. We started off with an idea. So we started off with nothing. We'd come out of Arrow Therapeutics. We were looking at a target and how we might actually deal with the target area. We looked at properties of compounds associated with RSV fusion at that time, and it's shown in this graph in the top left. And basically the major property of this was that compounds tended to be getting to be bigger, higher log Ps, and the issues associated with that, higher lipophilicities, fatty molecules, molecules that could have developability issues, solubility issues. And so what we decided to do was to target molecular changes that would make those compounds lower log P, more acceptable, and perhaps with greater potential for moving forward once those compounds got into a developable situation. And our first stage was literally to prepare our screening set. Basically, at this point in time, we were using money put together by the founders. We had £55,000 to do this with, and with that money, we made 30 compounds. 30 compounds which explored the structural modification and dealt with these physical properties as best as we could. What was really good for this is that what came out of this was a range of compounds. You can see in this plot, we're actually plotting the fusion PIC50. So basically the compounds get more potent the further up we go against the C log P. And you can see that our compounds tended to spread. So yeah, we did make some compounds with high log P. Being a chemist, they were the easiest ones to make. So obviously we made those to get started. But as we managed to drop the log P with this structural modification, most interestingly, our most potent compounds sat in the top left-hand part of this graph. Most potent compounds with actually the lowest log Ps. This gives you a structural angle on what these compounds were. We picked the benzimidazole right-hand component and varied the left-hand component of this molecule. We looked at 6 5 systems and 6 6 systems. Now, what was, I suppose, as a chemist, synthetically useful at this point was the actually harder to make 6 6 systems all failed quite poorly, really. Activities were poorer, and also we found their log Ps hard to control. The 6 5 smaller systems seem to be more potent generally, sitting below a micromolar. And our best compound, 49 here, RV521, was actually about 30 nanomolar. And it's actually one of the smallest molecules in the group. Quite related to it is this is a compound which actually was about the same sort of figure. Well, about 10-fold back, but pretty good. 49, we took forward into mouse in vivo pharmacokinetics, and also did some ADME studies in vitro. Our log P was a little bit high for this. We had an interesting kinetic solubility, and we had a permeable compound. And interestingly for this compound, we probably got higher protein binding across the range of protein binding studies than we probably expected, quite frankly. The downside was a bioavailability of 4%. That was a bit of a blow, but on the other hand, we could have ended up with absolutely nothing. What this compound allowed us to do was to put a proposal forward to the Wellcome Seeding Drug Discovery Initiative. And on the back of this, you could call them foolhardy, I don't know, but on the back of it, they funded the project and actually funded another £3 million into this project, which was a great boost. I mean, to be perfectly honest, at this point, we'd gone as far as we could with our money. We'd spent everything, we'd made the compounds, we had the compounds we had. Having got the money in place, the next stage was to actually have a look at the issues associated with that compound, what was affecting its permeabilities and its general appearance, if you like, in vivo. We ran a metabolite ID study. It pointed very much to the fact that this part of the compound, this cyclopropyloxindole, was the target for metabolizing enzymes. And when we screened these compounds in fusion and plaque assays, the 6-fluoro compound was actually well ahead of other compounds, with the possible exception of the 5. The 5, it has to be said, is synthetically quite a taxing molecule. It doesn't really matter how you construct this part left-hand end of the molecule, you get the 5 compound out as a minor component. So our attention for very much that reason, with what looked like some improved stability, focused on the 6-fluoro. Here's some data on the 6 and 7-fluoro. You can see that we're retaining plasma protein binding. We've got pretty decent solubility. It's actually very good in the case of 7-fluoro. Our permeabilities, we've now changed assay as well, which I do apologize for, but we moved contractor when we went into this stage of the process, having got more money in. And we showed that this 0.75 figure in this permeability assay is really quite a good one. It tends to suggest you're going to get some sort of oral absorption, and this was quite obvious, where we're seeing a 25% bioavailability for the 6-fluoro, which dropped drastically with the lower permeable compound. So that was where we got to. We got a 6-fluoro compound, which looked like we got a step forward. We'd improved oral bioavailability. We turned our attention to the chain off the bottom of the benzimidazole. We had some metabolite ID work, which suggested this was also a target. And we made a range of chains off this benzimidazole, some very good ones, actually, all looking at dropping log P or log D in this case, since we've got a basic center in the molecule. And we could see that these were dropping over the neopentyl. So if the R was the alkyl group we saw in the previous molecule, so this sort of neopentyl group, this chain actually drops the log P further. And trifluorobutyl looked really good from all sorts of angles. It was certainly our most active compound, both in fusion and plaque. It actually had a very good polar surface area, which again is, the more we're increasing our polar surface area, the more we're reducing the absorbability, the oral absorbability of the compounds. And with this compound we called RV521. That came a little bit later, actually. But looking at this compound in vivo and in vitro PK, in a mouse, we're seeing really good exposure with the compound, despite what was a moderate clearance figure actually still being present well above, in this case, plasma-adjusted EC90 at 8 hours. The protein binding is still pretty good. Permeability is acceptable. That's reflected in the oral availability. Microsomal half-lives are actually supported and improved by hepatocyte half-lives, where we're seeing quite long half-lives, actually. Clearance figures are still moderate, but actually, looking at the profile of the compound, we're seeing a good oral exposure at 10 mg per kg in mouse. We're well above the plasma-adjusted IC90, which in this case, we're probably 3 or 4 times above that at 8 hours. We progressed the compound into an animal model. We ran the BALB-C mouse model. There were a couple of animal models of RSV infection. Cotton rat is another which is frequently used. There's plenty of data around about compounds of this type in the BALB-C mouse model, and we followed that. What we showed by treating at T-2 hours with RSV521, then infecting with RSV at T0, we could, and then subsequently treating at 24 hours, we saw significant reductions in viral load. So as a percentage of the control, even at 10 mg per kg, we were dropping the viral load quite significantly. 50 mg, it almost disappears, but I mean, most things should drop everything at 50 mg per kg, quite frankly. It's a huge dose in animals. We also took the compound in the human airway epithelial model. This is an in vitro system that displays the morphology of the human airway epithelial, actually in a Petri dish, where you can dose with virus topically and basolaterally you can dose with compounds. So you can sort of mimic systemic exposure passage through epithelial membranes of a compound as well. And we tested this compound at 10, 100 and a nanomolar and a micromolar, and against a positive control, which was an earlier BMS compound, in this case, dosed at a micromolar. And we saw that this compound effectively reduced viral titers. This is based on luminescence figures from luciferase-linked RSV, recombinant RSV, and virus shown in this case is determined at day 7 and day 8. And you can see that the viral load by this measure was reduced significantly over that period of time with 2 doses. RSV being what it is, we spread the non-specificity of treating RSV across A and B subtypes and found that we got a range of A and B subtypes inhibited, some significantly below a nanomolar, all below 10 nanomolar. We saw across a range of cell lines toxicity all in the micromolar scales, so 1,000-fold plus back for this compound. Moving the compound into rat and dog, we saw good exposures in both species, which essentially allowed us to start to predict absorbable dose in man, at this case, and to scale to man. RSV fusion inhibitors have a track record, if that's what you want to call it, in resistance generation. We looked at generating resistance mutants to RV521 in the Memphis 37B strain over 10 passages. It took us 10 passages to produce that resistant mutation. It didn't appear in passage 1. We identified a single mutation, D489Y, in the RSVF protein. It showed cross-resistance with competitor compounds like GS5806. You would expect that. The Gilead compound showed a larger EC50 shift than 521 against the variants we prepared, similarly for J&J678. And the RSV mutant remained sensitive to other replication inhibitors, either RSV604, which came from Arrow, or the allelose nucleoside inhibitor. This is a quick, docking study, molecular modeling study. It's not a crystallography, so it's got some worth. What it actually shows is what we would perceive to be an acceptable docking mode of RV521 to the trimeric complex at that center binding point I described earlier. What it actually does, which shouldn't be any surprise to anybody really, is put the aminomethylene function into an acidic portion of that trimeric structure where you've got multiple acidic residues, either aspartic acid 486 or threonine 397, serine 398, and aspartic acid 489 isn't too far away, probably about an angstrom further out than those other residues. So it's a reasonable binding mode. It's a binding mode which puts the molecule fairly close to its mutatable residue, fairly close, and all perfectly reasonable. I wouldn't put my house on it, though. It's a modeling study.
I think it'd be wise to move forward to the...
Sorry, yeah.
We've got 3 more minutes left. So you can with the clinic, if you can.
You don't need to know about synthesis. We've been able to prepare it on multiple kilo scale. The compounds progressed through preclinical evaluation. GLP safety studies, no effect with regard to AIMS, mutagenicity, cardiac liability, broad pharmacological profiling. Preclinical tox package has been completed and it's moved into a randomized double-blinded phase 2A clinical study in healthy adult volunteers. So this is the usual healthy adult volunteer clinical study. RSV inoculation. Patients are then tested for whether they are RSV positive or not. Subsequently, days 2, 3, 4, 5, and dosing of the compound or placebo follows subsequently to those patients being viewed as being positive for RSV. And participants received either 200 mg or 350 mg or placebo twice daily for 5 days. What we found was this compound significantly reduces RSV viral load. So quantitative PCR, the data falls off pretty quickly at both 200 and 350 mg. If we're looking at assay RSV measured by plaque assay, we see that it still falls off pretty quickly. It's pretty significant. It's actually, from what I can tell, comparable to anything Gilead saw with their compound and is probably significantly better than anything Johnson & Johnson saw with theirs. The viral load is repeated in clinical symptoms, so a total symptom score shows a very quick reduction of symptom score, and mucus weights, which was a secondary endpoint for the phase 2A study, never really appear, quite frankly. So what we've got is a potent inhibitor of the RSV fusion process, good solubility, good PK in mice, rats, and dogs, good safety profile, no serious adverse events reported in either phase 1 or phase 2 studies, and phase 2 study adverse events were reversible and actually associated with a fasting process for patients. So if you didn't eat for a day and then somebody chucked some gunk down your mouth, you would probably not feel very well. It's that sort of thing. Only a single RSV genetic variant detected following RSV521 treatment. That's going to be the subject of future publications. And there's no effect of clinical resistance or viral load rebound associated with the compound. It just behoves me really to finish by referring to people who supported this program. There's a group of people over the years, quite frankly, who supported Reviral practically. Some of these people are still with the company, some people have moved on. The initial founders all came from Reviral, all came from Arrow, Surrey, and supported financially the company, myself included. So that's my disclosure. And contractors. We used a large range of contractors in this program, from Paul Young at Queensland through to contract companies in the UK. And we continue to and have expanded this. So Reviral is very much a semi-virtual company. It's got a basic virology and core medicinal chemistry expertise, but makes full use of a large range of contracting companies to support its programs. That's me done.
Thank you.